Taste perception begins with specialized receptors on the tongue, each tuned to detect one of 5 primary categories: sweet, salty, sour, bitter, and umami. These receptors are not uniformly distributed, but are localized in specific regions, allowing the tongue to sample a wide variety of flavors simultaneously. Sweet receptors signal the presence of carbohydrates, providing a rapid cue for energy-dense foods. Salty receptors detect sodium, a critical electrolyte for neural and muscular function. Sour and bitter receptors often serve as protective mechanisms, warning the body of potentially harmful acids or toxins. Umami receptors respond to glutamate, indicating protein content and nutritional value. The activation of these receptors is the first step in a complex sensory and neural cascade that determines whether a food will be perceived as desirable or aversive. Once activated, taste receptors transmit signals via cranial nerves to the brainstem and then to higher-order sensory centers, including the gustatory cortex, which processes and integrates these inputs. However, taste perception is far more than a simple reading of chemical signals. The brain constructs flavor by combining taste receptor input with information from other sensory modalities. Olfactory signals, or smell, are particularly influential, allowing the brain to detect volatile compounds that contribute to aroma and enhance the perception of flavor. Similarly, texture and temperature are integrated into the overall sensory experience, with crunchy, creamy, hot, or cold textures influencing the appeal and palatability of food. Even visual cues such as color and presentation can modulate flavor perception by setting expectations that prime the brain for particular taste experiences. This integration is rapid and automatic, creating a holistic representation of flavor that is both sensory and emotional. The brain does not process taste in isolation. It simultaneously evaluates the potential reward or threat value of the food. Sweet or umami flavors often trigger circuits associated with reward, while bitter or sour flavors may activate avoidance or cautionary signals. These early neural evaluations contribute to the initial attraction or aversion experienced when encountering food and form the foundation for future taste preferences and cravings. The sensitivity of taste receptors can also vary based on genetic factors, age, and experience. Some individuals have heightened sensitivity to bitter compounds, influencing their avoidance of certain vegetables or coffee, while others may have a higher threshold for sweetness, driving preference for more sugar-rich foods. Repeated exposure can modify these thresholds over time, as the brain learns to associate particular sensory experiences with positive or negative outcomes. For example, consistent consumption of a particular fruit may enhance its perceived sweetness, even if the chemical composition remains constant, demonstrating the brain's ability to adapt taste perception based on experience. Furthermore, the brain links taste to memory and emotion, forming associative networks that influence future food-seeking behavior. The pleasure derived from a favorite meal is stored as a neural pattern that can trigger anticipatory cravings when similar sensory cues are encountered. This connection between taste, reward, and memory explains why certain foods evoke strong emotional responses or nostalgia. Understanding the biology of taste receptors and flavor processing highlights that cravings and food preferences are deeply rooted in sensory perception and neural integration. These mechanisms provide the foundation for why some foods are irresistibly appealing and others are avoided, demonstrating that taste is not a simple sensory event, but a complex interaction of chemical, neural, and cognitive factors. The brain's reward system is a central driver of taste cravings, linking the sensory experience of food to motivation, learning, and future behavior. When a highly palatable food is consumed, specific neural circuits release dopamine, a neuromodulator that signals reward and reinforces behaviors associated with pleasure. This system evolved to ensure survival, encouraging the consumption of energy-dense foods in environments where calories were scarce. Today, this same system responds to modern foods that are high in sugar, fat, and salt, producing intense motivational signals that are difficult to resist. Dopamine does not directly create the sensation of pleasure. Rather, it enhances the salience of a stimulus, making the brain prioritize seeking out the rewarding food again in the future. The mesolimbic pathway, which connects the ventral tegmental area to the nucleus accumbens, plays a critical role in this process. When activated by food consumption or even the anticipation of food, these regions increase dopamine signaling, reinforcing the connection between the sensory properties of the food and the behavioral response to obtain it. Over time, this reinforcement strengthens neural pathways, creating habitual patterns of seeking and consuming certain foods. Environmental cues, such as the sight, smell, or even thought of a food, can trigger these reward circuits in anticipation, producing cravings before any calories have been ingested. This explains why just walking past a bakery or smelling a freshly baked treat can create a strong desire to eat, even in the absence of hunger. Learning and memory are deeply integrated with the brain's reward system, further influencing cravings. The hippocampus and amygdala interact with reward circuits to store experiences associated with specific foods. If a particular food is consistently paired with a positive emotional state, such as social connection, stress relief, or comfort, the brain encodes this association. Later, encountering the food or a similar sensory cue activates not only the reward system, but also the emotional memory networks, amplifying the craving and creating a strong motivation to repeat the behavior. This is why comfort foods, snacks consumed during celebration, or meals linked to positive experiences are often craved more intensely than neutral foods. The intensity of reward signaling is modulated by metabolic and hormonal states. Hunger increases the sensitivity of reward circuits to food-related stimuli. Ghrelin, a hormone released when the stomach is empty, enhances dopamine activity in response to food, making energy-dense foods appear more appealing. Conversely, satiety and leptin signaling from fat stores can reduce the activation of these circuits, lowering the perceived reward value. Chronic overconsumption of highly palatable foods can, however, impair this regulation, leading to a situation in which the brain requires more stimulation to achieve the same reward response. This recalibration contributes to stronger cravings and may drive overeating. Reward circuits are not fixed and can be reshaped over time. Repeated exposure to certain foods strengthens the association between taste and reward, while reducing exposure or introducing alternative, nutrient-rich foods can gradually recalibrate the system. Mindful eating practices, which involve attention to flavor, texture, and internal signals of hunger and fullness, also modulate reward signaling by enhancing cortical regulation over subcortical reward pathways. This demonstrates that while the brain's reward system strongly drives cravings, it is not immutable. Intentional strategies can influence its responsiveness, guiding food preferences toward healthier patterns. Hunger hormones play a fundamental role in shaping taste cravings and guiding food-seeking behavior. Two of the most influential hormones in this process are ghrelin and leptin, which provide the brain with real-time information about energy status and influence both appetite and reward circuits. Ghrelin, often referred to as the hunger hormone, is released by the stomach when it is empty, signaling the brain that energy intake is required. This hormone not only stimulates the sensation of hunger but also sensitizes the brain's reward system, making energy-dense foods appear more desirable. When ghrelin levels rise, dopamine release in response to food-related cues is amplified, increasing both motivation and the intensity of cravings. This effect explains why foods high in sugar, fat, or salt can feel almost irresistible when the stomach is empty or during periods of prolonged fasting. Leptin, in contrast, acts as a satiety signal. Secreted by fat cells, leptin communicates to the hypothalamus and other brain regions that energy stores are sufficient, helping to suppress appetite and reduce the drive to seek food. When leptin signaling is strong, reward circuits respond less intensely to highly palatable foods, allowing the brain to prioritize energy needs over hedonic desires. However, chronic overconsumption of calorie-dense foods, obesity, or metabolic disruption can lead to leptin resistance, in which the brain fails to adequately register energy sufficiency. Under these conditions, cravings remain high even when the body has excess energy, and food-seeking behavior becomes more strongly influenced by reward circuits than by actual metabolic need. Other hormones also contribute to the modulation of taste cravings. Insulin, for example, helps regulate blood glucose levels and signals nutrient availability to the brain. Sharp fluctuations in blood glucose can trigger cravings for simple carbohydrates or sugary foods as the body seeks to restore energy balance. Cortisol, a hormone released in response to stress, can increase cravings for high-calorie foods, particularly those that are sweet or fatty. This effect is thought to be an adaptive response, preparing the body to quickly replenish energy stores in the face of perceived threats. Over time, chronic stress can amplify this process, making reward-driven eating a common coping mechanism. The interaction between these hormones and sensory inputs is critical for understanding how cravings emerge. Ghrelin, leptin, insulin, and cortisol all influence the sensitivity of the brain's reward system to taste, smell, and texture cues. When ghrelin levels are high, even subtle sensory signals from the environment, such as the aroma of baked goods or the sight of a snack, can produce strong anticipatory dopamine responses. Conversely, when leptin signaling is effective, these cues are less likely to trigger intense craving, demonstrating that the hormonal state of the body directly shapes the perception of food's desirability. Beyond short-term regulation, hormonal signals also interact with long-term learning and memory to shape preferences. Foods consistently paired with periods of high ghrelin or stress become strongly encoded in reward circuits, increasing the likelihood of future cravings under similar conditions. This process illustrates why habitual eating patterns often persist despite conscious intentions and underscores the importance of aligning physiological states, dietary choices, and environmental cues to manage cravings effectively. Learned associations and emotional links play a central role in shaping taste cravings, demonstrating that the desire for specific foods is not purely physiological, but also deeply tied to experience and memory. From a neurological perspective, the brain continuously forms connections between sensory inputs, emotional states, and behavioral outcomes. When a particular food is consistently paired with a positive experience, such as social bonding, stress relief, or comfort, the brain encodes this relationship in circuits connecting the reward system, memory centers like the hippocampus, and emotional processing regions such as the amygdala. Over time, encountering sensory cues associated with that food, whether it is the smell, taste, texture, or even visual presentation, can trigger anticipatory neural activation in reward pathways, creating cravings that occur independently of actual energy needs. Emotional states are particularly influential in reinforcing these learned associations. Stress, boredom, and excitement can all modulate how the brain links food to reward. For example, consuming a sweet or high-fat snack during periods of stress activates the reward system in a way that temporarily alleviates negative emotions. The brain then encodes both the emotional relief and the food's sensory characteristics, increasing the likelihood that the same food will be sought in future stress-related contexts. Over time, this process creates a feedback loop in which specific emotional states automatically trigger cravings for certain foods, even when there is no physiological hunger. Memory also amplifies these associations. Positive experiences related to taste, such as family meals, celebrations, or pleasurable restaurant visits, are stored in long-term memory and can elicit anticipatory cravings when similar contexts arise. This anticipatory response is mediated by the prefrontal cortex and the hippocampus, which retrieve contextual and episodic information that primes the brain's reward system. In other words, just thinking about a food that has been consistently linked with pleasure can trigger neural and hormonal responses akin to actually consuming it, creating a strong motivational signal to seek it out. The process of conditioning extends beyond conscious experiences to include subtle environmental cues. The sight of a particular snack on a store shelf, the smell of baking bread, or even specific times of day can serve as triggers for cravings. Because these stimuli have become associated with prior rewarding outcomes, classical conditioning mechanisms explain how these cues acquire predictive value, signaling the availability of a rewarding food and activating dopamine-mediated circuits even before the food is encountered. This explains why cravings often occur in consistent patterns, such as desiring dessert after dinner or feeling drawn to sugary snacks in the mid-afternoon. Learned associations can also create aversions, highlighting the bidirectional influence of experience on taste preference. If a food is paired with a negative emotional or physical outcome, such as nausea, disgust, or social embarrassment, the brain can encode avoidance pathways that decrease future consumption. These aversive memories engage circuits that inhibit reward signaling, demonstrating the flexibility of learned associations in shaping eating behavior. Over time, the interplay between learned associations, emotional context, and environmental cues can dominate eating behavior, often overriding physiological signals of hunger and satiety. Understanding the mechanisms of these learned links provides insight into why cravings can feel automatic and compelling and why strategies to modify them, such as altering context, introducing new associations, or practicing mindful awareness, are necessary for regulating intake. The gut microbiome plays a surprisingly influential role in shaping taste cravings, highlighting that food preferences are not determined solely by the brain or sensory systems, but also by the trillions of microorganisms residing in the digestive tract. These microbes interact with the nervous system, endocrine system, and immune system, forming a complex, bidirectional communication network often referred to as the gut-brain axis. Certain microbial populations can influence the desire for specific nutrients by producing signaling molecules that affect appetite regulation, reward processing, and metabolic function. This means that the composition and activity of the gut microbiome can directly modulate cravings for sugar, fat, salt, or other nutrients based on the needs or preferences of the microbial community itself. Microbes in the gut can produce neuroactive compounds such as short-chain fatty acids, neurotransmitters, and metabolites that communicate with the central nervous system via the vagus nerve and through hormonal signaling. For example, some bacteria produce metabolites that stimulate appetite, while others can generate compounds that suppress hunger or enhance satiety signals. These microbial signals interact with key appetite-regulating hormones, including ghrelin and leptin, altering how the brain interprets energy needs and how strongly it responds to food-related cues. The net effect is that certain microbial profiles may predispose an individual to seek out calorie-dense or nutrient-specific foods, while other profiles may reduce cravings and promote dietary balance. Research suggests that gut microbes can also influence reward pathways in the brain. The same dopamine circuits that respond to highly palatable foods are modulated by microbial signals, creating a situation in which the microbiome can bias taste preference and the motivational salience of certain foods. For example, when a particular microbial population thrives on sugar, the brain may become more responsive to sweet flavors, reinforcing the consumption of sugar-rich foods. Conversely, when fiber-digesting microbes dominate, the brain may respond more favorably to complex carbohydrates and plant-based foods, demonstrating a form of microbial-driven taste adaptation. The gut microbiome is highly dynamic and responds rapidly to dietary patterns. A diet high in sugar, fat, and processed foods can favor microbes that reinforce cravings for those same foods, creating a feedback loop that promotes habitual consumption. Similarly, diets rich in fiber, vegetables, and whole foods can encourage microbial populations that support reduced cravings for highly processed foods and enhance the reward value of nutrient-dense options. This demonstrates that taste preferences are not fixed, but can be gradually recalibrated through dietary interventions that reshape the microbial ecosystem. Early life microbial exposure and long-term lifestyle factors also influence how strongly the microbiome affects taste behavior. Birth method, antibiotic use, stress, and diet in infancy can shape microbial diversity, which in turn affects metabolic signaling and food-seeking behavior later in life. Microbes interact with immune cells and endocrine pathways to influence inflammatory states and metabolic efficiency, further modulating the brain's responsiveness to food and the intensity of cravings. Attention and conscious dietary choice can modulate these microbial influences over time. Repeated exposure to certain foods alters microbial composition, which gradually changes taste perception and craving patterns. Mindful eating practices, by slowing consumption and increasing awareness of sensory experience, can also reduce the dominance of microbial-driven reward signals by allowing the brain to integrate physiological and cognitive cues before rest lifestyle factors and sensory adaptation play a pivotal role in shaping taste preferences and cravings, illustrating that the intensity and direction of food desire are influenced not only by biology, but also by habitual patterns and environmental conditions. Sleep, stress, and diet quality directly affect hormonal signals such as ghrelin, leptin, and cortisol, which in turn influence the brain's reward circuits and sensitivity to food cues. Sleep deprivation, for example, has been shown to elevate ghrelin levels while reducing leptin, creating a physiological state in which the body signals hunger more intensely and the brain perceives foods, particularly those high in sugar and fat, as more rewarding. Chronic stress similarly activates cortisol pathways, which amplify cravings for calorie-dense foods as a means of energy mobilization and emotional regulation, demonstrating that lifestyle conditions can profoundly modulate the biological drivers of food-seeking behavior. Sensory adaptation further shapes how cravings are experienced over time. The brain's reward circuits respond not only to the intrinsic properties of food, but also to previous exposure and frequency of consumption. Regular intake of highly processed, sugar-rich, or salt-heavy foods can recalibrate the brain's reward system, increasing the threshold required to achieve the same level of satisfaction. This process explains why naturally sweet or complex foods may initially seem less appealing after prolonged exposure to hyperpalatable diets. Conversely, reducing exposure to these intense stimuli allows taste receptors and reward pathways to gradually readjust, enhancing the perceived palatability of whole foods and nutrient-dense options. The principle of adaptation demonstrates that taste preferences are flexible and that conscious modulation of sensory experience can recalibrate cravings over weeks or months. Attention and mindful engagement with food can accelerate this recalibration. Actively observing the flavors, textures, and aromas of each bite increases cortical awareness of sensory input, allowing higher-order brain regions to modulate reward responses more effectively. This conscious focus interrupts automatic, habitual consumption patterns and increases the ability to experience satisfaction from subtler, more natural flavors. Over time, these practices strengthen neural circuits associated with controlled intentional eating and reduce the dominance of hyperpalatable cues that previously drove cravings. Environmental and social contexts also influence sensory adaptation and cravings. The visual presentation of food, color contrasts, portion sizes, and even cultural associations can prime the brain to anticipate reward. Exposure to consistent environmental cues, such as always consuming a sugary snack while watching television, reinforces the learned association between stimulus and reward. By altering the environment or breaking habitual pairings, the brain can gradually weaken these conditioned responses, reducing the automatic drive towards specific foods. Dietary variety and timing play additional roles in shaping taste perception. Consuming a wide range of flavors, textures, and nutrient profiles can enhance neural responsiveness to different foods, preventing sensory monotony and promoting more balanced nutrient intake. Regular meal timing and spacing of snacks also stabilize metabolic and hormonal signals, reducing exaggerated swings in hunger and minimizing opportunistic cravings that arise from physiological imbalance. Through the integration of lifestyle factors, sensory adaptation, and conscious behavioral strategies, the brain's response to taste can be recalibrated. This dynamic interplay demonstrates that cravings are not fixed impulses, but modifiable outputs of neural, hormonal, and sensory systems interacting over time. On understanding the biology of taste cravings reveals that they are not random or purely a matter of willpower, but emerge from a complex interaction of sensory processing, reward circuits, hormones, learned associations, gut microbiome signals, and lifestyle factors. By increasing awareness of these mechanisms and making deliberate adjustments, such as modifying diet patterns, practicing mindful eating, managing stress and sleep, and reshaping sensory exposure, individuals can recalibrate their brain and body's response to food. Over time, this allows cravings to align more closely with actual physiological needs rather than habitual or reward-driven impulses, fostering greater control over eating behavior, improved nutrition, and long-term health.